So this is the Whole Genome Sequencing for Cluster Detection and Investigation Live Learning series. We've done these a few years now through the Tennessee Integrated Food Safety Center of Excellence. This particular series will have 3 sessions. And at the very end of today, I'll kind of go through what the next 2 are. This first one will be kind of just WGS fundamentals and lead us into the next 2 sessions. So I am Lauren Hudson. I'm a research assistant professor in the Department of Food Science at the University of Tennessee. I've been working with TDH and the Tennessee COE for over 5 years now. So I've been doing this for a little while. So I come from an academic background, not public health background. So it's a little bit different, but I understand at least the science part of things. So we'll have this overview. We'll do some WGS basics about how sequencing is actually performed. We'll go into SNP analyses, core genome MLST and whole genome MLST base analyses, looking at phylogenetic trees or dendrograms and SNP or allele matrices. Then since this is an 1.5-hour session, we'll do like a 5-minute break in the middle to refresh. And then in the second part, we'll go into allele codes, X codes, using thresholds for cluster detection. And like I said, we'll kind of talk through what the next couple sessions are and have some time for questions or discussion. And just kind of to give you like a feeling of what I'm hoping for you all to get out of this is that I know most of you actually are epidemiologists. So I want to empower you all to feel more confident with WGS and interpreting WGS results, and even taking the reins some and maybe running your own analyses or something like that. I don't want you to feel that you are only able to get the results that are interpreted by someone else. I want you to feel empowered to be able to interpret things yourself and really understand what they mean, and so that you can apply this to your work. Okay, so start off, we'll go a little bit into the science of the actual sequencing process. So it'll start, usually at the public health lab. They'll have a bacterial culture they'll receive or grow up, and they'll take the bacteria from this plate and they'll treat them with something that will break the cells open, lyse the cells open, and release the DNA from the cells. The DNA will be purified. The DNA, while it's still in these big chunks, it'll be cut into shorter fragments using either enzymes or mechanically broken up into pieces. And then the library preparation will happen. So it's basically getting everything set up for the sequencing. And the DNA that goes into the sequencer is called the DNA library. So sometimes you'll have to add adapters depending on the sequencing process it's going into and things like that. And then finally, this is kind of what people think of when we think of the sequencing. It gets loaded onto the sequencer, and the sequencer will read the ATCG nucleotides at each position, and it'll determine those. And then all of that data can then go into a computer and be further processed by other programs or into different systems. So just a little bit more in depth with Illumina sequencing, which is what we're mainly doing in public health, at least right now. The DNA fragments are going to be pretty tiny. I think they're usually like 150 to maybe 300 base pairs. So we end up with all of these little chunks. And even with the sequencing, when we get that result, it's going to be these little sections of sequences. And depending on what we need to do with that, we may try to, it's kind of like a puzzle pieces. You may try to put them back together to kind of be the entire genome, or you may just have little sections that are bigger things called contigs. But it's more difficult to put them back together into an entire genome with a short read sequencing like we do in public health. But again, for some purposes, we don't even need to do that. So after the sequencing, they'll go through quality control. So like with PulseNet, they have quality metrics they have to meet. And so things sometimes might need to be sequenced again. And then, like I said, we may or may not need to assemble the reads into an entire genome. Depends on what we're doing with it. And then the main thing is we do want to do variant calling, especially like for these public health surveillance applications. So there's 2 main types we'll talk about. So we have single nucleotide polymorphism, or SNP-based ones. And then we have gene-based ones that include whole genome or core genome MLST. And based on those results, we can subtype our isolates or determine how closely related they are based on that. First off, I'm going to talk about SNP-based analyses. I think it's a little bit easier to understand. So we're basically going to use those single nucleotide changes to infer the relatedness between isolates. And we can look at the entire phylogeny of a group of them, and it can also be used for strain typing. And then these single nucleotide changes are typically going to be things like standard mutations that happen over time in bacteria when there's a mistake in the processing. So to do that, we're going to map those raw reads, like I showed you, to a reference genome. Again, it's kind of like little puzzle pieces. We're going to map them back where they go to a reference genome. And then the program would go through and identify the SNPs, so places where the nucleotides do not match up to that reference. So in this example down here is just a little snippet of it, but if the reference sequence is ATGTTC, here, and then our sequence here would be ATGTTG. So right here in this position, that would be considered a SNP because the nucleotides are different. And then some programs will go through and remove lower quality SNPs because, like I said, we're mapping them. I'll show you a picture in the next slide. And the sequencers aren't perfect, so the program may have some kind of quality threshold to be able to call it a SNP or not. And then from that, we can create a multi-sequence alignment. So basically, we just, instead of having all of the sequences, we just take out the SNP positions. And then we can use that to infer the phylogeny. So here's kind of just a simple illustration of all of this. So here is this blue line here would be like the reference genome that we're mapping the reads to. And then these little thin rectangle things here are going to be our reads, right? So we're mapping them back to the reference. And again, we'll have a bunch of little reads and they'll map to where they need to go in the reference. And in this example, we have 3 different isolates, isolate A, isolate B, and isolate C. So now that they've been mapped, the program would go through and look through at this, and then it finds that this position, isolate A and isolate B, both have an A in that position, but isolate C has a T. So that would be a SNP. Here's a position. It's not a SNP because they are actually all the same, but I kept it there to illustrate. But so this would not be a SNP because they all have the same nucleotide there. And then this one, we have 2 Gs and a C, so that's a SNP. And then this one, we have an A, a T, and a T. So if we want to use that to compare these isolates to each other, we can extract those SNPs out and look at them lined up. So again, we have ACGA for isolate A. We have ACGT for isolate B. And then we have TCCT for isolate C. So if we look at those now, we can say that isolate A and B are more closely related than isolate A and C. And we know that because if we compare isolate A and B, there's only 1 SNP difference. But if we compare isolate A and C, we have 3 SNP differences. So that means they are more distantly related. So here's that multifaceted alignment I was talking about. So basically this just extracted all the SNP positions out. And so you can see all the SNPs are lined up, and you can tell in certain columns where, like this one here, there's some T's and A's, but for most of the columns, a lot of the things are the same. And from that, we can make a SNP matrix, which basically is just going to give you the differences between 2 different isolates. And I will show you how to read that. The 2 main ways you'll usually see these types of results are on a phylogenetic tree or a SNP matrix. So the SNP matrix is set up where all of the isolates you're looking at are going to be in the vertical column or in the first horizontal column. And if you want to know the difference between 2 isolates, you would just follow it along into the 2 you want to look up are where they intersect. I will show an example of that in a minute. And then we'll look at also how to look at this stuff on a tree. So when you're looking at a tree, I just want to kind of point out some things. The root would be where a tree starts. The branches are these horizontal lines throughout the tree. And then there's things called nodes. That's kind of where they meet. So there's internal nodes throughout the tree, and then these leaf tip or external nodes here on the ends. And then typically you have leaf labels that correspond to whatever your isolate names are, or they may be maybe P and USA numbers, whatever you're looking at. And two important things. A tree, if you're looking at it, it should have a scale bar usually. If it doesn't, that may be suspect. So it'll have a scale bar and it'll tell you what the branch lengths actually mean. And so like in this one, this length here is going to be about 5 SNPs. And if it doesn't have a scale bar, it'll usually have the branch lengths above the branches instead. So it'll usually have one or the other, sometimes both. And the other important thing is knowing that the direction of evolution is going to be horizontal on a tree. So we could stretch this tree out vertically and it's not going to change anything. But if we stretch it out and mess up the distances that are horizontal, that's going to change the way we interpret this tree. Okay, so let's look at some examples looking at this tree. So let's say that I want to know the difference, the SNP difference between isolate G and isolate H. So we could look at that up on the SNP matrix. So I find the G row and find where it meets with the H column. And then we got 0, so 0 SNPs. And then we can also look that up in the tree. Basically, you just want to follow the path from G to H on the tree. There's actually no horizontal distance between G and H, so the SNP distance is also going to be 0 from there as well. And then the matrix, it works either way. So like I could have gone to the H row and saw where that met with the G column. They should be the same. A lot of times with the matrices, you might only see the lower half or the upper half. And also with the matrices, you'll notice that the diagonal line through it will always be 0 typically. And that's because that's where you're comparing each isolate to itself. So if like, let's go see here, we'll go D to D. When it's compared to itself, it's going to be 0 SNPs because it's the same thing. Okay, so let's look at another one. If we want to know the difference between G and I. So let's see, find the row for G and the column for I. So the matrix tells us it's 3 SNPs. And if we want to do that on the tree, again, like I said, we would just kind of follow from G to I. So let's see here. And then we go along this branch, and that branch is 2 SNPs in length. And then we go down, and then we follow this branch, and it is 1 SNP in length. So to get to I, it took 2 plus 1 is 3. So it's going to be 3 SNPs. So we got the same result from both. Okay, so I have a poll I am going to put up. So everyone try to answer this question. You can use the matrix and or the tree to look up the answer. Okay, like 5 more seconds. Okay, so it looks like almost 94% of people got that right. So great job. The answer is actually going to be 0 SNPs. And that's, like I said, most of y'all got that. So if we looked on the matrix A and C where they meet, 0. And then A to C here, there's no horizontal distance between the two. So also 0. Let's try a different one. It's a little bit more complicated. And 10 more seconds. Okay. Again, I am super impressed. 95% of y'all got that right. Um, so the SNP distance is going to be 8 SNPs here. And again, if we look at the A row and then where it meets the D column, we get 8. And then if we want to look on the tree, we follow from A, go down, go over 5 SNPs, and then we go over 3 to get to the D. So 5 plus 3, that is 8. 8 SNPs. And then I just wanted to like go a little bit. I don't want to go too far into the weeds with this, but this is one of those things where it seems like there's a lot of different terms people use. People use them very interchangeably. So I just want to like go through it a little bit. So we often hear the word dendrogram. A dendrogram is a very more broad term. It's not just phylogenetic trees or genetic stuff. It could be any sort of clustering type, tree type diagram, right? So it's a broad term, and if you don't know what to call something, a dendrogram is probably a safe word to use. But yeah, so it's just going to be like for any tree-like diagram. And then so phylogenetic trees would fall into that, and it's going to be a general term for a diagram representing evolutionary relationships. And so you may also hear cladogram or phylogram as well. Cladograms are usually going to be unscaled, so the branch lengths are arbitrary, so we couldn't use the branch lengths like we just did to determine the relatedness, but you could kind of look at how they branch and determine how things are related, but not by how much, maybe. And then a phylogram is going to be scaled, so it does. So that's kind of the trees we were looking at, where the branch lengths do represent amount of change, but it may not show exactly the time. Like we looked at the SNPs, right? We don't know what kind of time period it was. And something you may hear about, I don't see them very often, but chronograms are basically like phylogenetic trees, but the length is indicating time instead. And then one more thing I wanted to show y'all while we're still talking about trees is that sometimes you'll see really different trees. They're all mean the same thing usually, but they just are kind of set up differently. This rectangular style one is like the one we just looked at. Sometimes they may be kind of more branch looking lines. Sometimes they could be curvy like this. And then more often I see these circular ones. So they usually take up less space. So that's one of the benefits people use them. But basically, you're reading it exactly like the rectangular version we saw, except this is going to be the root here in the middle. And then sometimes you'll see radiation or unrooted trees. And so these are not always great because you often end up with things overlapping on them, but these I find easy to see kind of where things branch off because of how it's set up. But yeah, so these all, are different types of trees you may see. So don't get scared if you see a different one. Just need to read it a little bit differently. So now we will move into the whole genome MLST and core genome MLST. For groups of organisms, let's say a species or a cerevar or a genus, there's going to be a core genome. So these are going to be genes that are present in all of the strains in that group. So for, let's say, Listeria monocytogenes, it would be genes that are present in all or almost all Listeria monocytogeneses. So like anything that's essential for them to be alive, things like that are definitely going to be in the core genome. And then there's going to be also an accessory genome. So it's genes that are present in only some strains. So that may be antibiotic resistance genes that they've picked up or other genes that not all strains in that species have. And together, those will make up pangenome. And so when we're doing core genome or whole genome LST, the core genome is only going to include the core genome, and the whole genome would include the core genome plus the accessory genome. And so if you want to think about it a little bit more in depth, so we tend to use core genome MLST for everything except for Listeria, where we use the whole genome MLST. Kind of a couple of reasons behind that I know of at least are because the whole genome MLST is more discriminatory. So that's one of the benefits of it. But it's less stable because the core genome, that's kind of a stable thing. Because like I said, it's genes that are in all organisms, but the accessory genome is going to be less stable because, it's continually adding new genes or new alleles in there. So yeah, there's some benefits and differences, but yeah, right now, whole genome LST is typically only used for Listeria, like for standard surveillance. For like example, with like Listeria, these numbers are kind of old, but it gets the point across at least. For the whole genome MLST scheme, it includes about 5,000 different loci or genes or areas. And then for the core genome MLST, it's about 1,800. So it's a lot smaller of a database of genes. And how this process worked is that all of the loci or genes are compared against a database of the known ones. And for each one, if it matches a known one or a version of it, like an allele, it is assigned an identifier. And if it does not match a known allele, it is going to be given a new identifier. So this database and scheme kind of grows as you go. And then all of the different loci or genes between the different genomes can be compared, and a distance is calculated and used to infer the phylogeny. So after that point, it kind of works like the SNPs. We look at the differences. So here's kind of a simple example of this. So here is isolate A, isolate B, and isolate C. And we're going to look at 4 different loci or genes. So those are each a different color. So the first one's blue, green, purple, and orange. So let's just look at this first one. So if these 3 genes are going to be compared, we look through and this one has a C here, this one has a C here, and this one has a G here. So these are going to be given the same allele identifier. So let's say gave it 1, and this one would be given a different allele identifier. It would be 2. And we're keeping track of them up here as the program goes. And the second gene here, this one has a GGA, this one has a CGA, and this one has a G, C, and T. So these are each going to be given a different identifier since they all have a different sequence in this gene. And something to note is that these would be counted as one allele difference, even though if you're comparing 1 and 3, there's actually 2 different SNPs or 2 different nucleotide differences. This would actually only be counted as 1 because we're only counting the alleles as a whole. Okay, and then for this purple gene, this one has a G here and a T here. This one also has a G here and a T here. So they're given the same allele identifier or designation. And then this one has a C and this one has an A. So it's going to get a different one. But again, if we're looking at differences later, that's only going to count as 1 difference, even if there's 2 differences within the gene. Okay, and this one, let's see, this one has a T, this one has an A, and this one has a T. So isolate A and C are going to get the designation of 1, and isolate B would get a designation of 2. And so now we kind of put that all back together like we did with the SNPs, and we have like the different allele designations lined up here at the top. So I'm actually going to go back a few slides. Yeah. So we have 1, 1, 1, 1, 1, 2, 1, 2, and 2, 3, 3, 1, based on the allele identifiers we gave each of these genes. And so similarly to before, we can determine which two are more closely related to each other. So this one, between isolate A and B, there is going to be 2 allele differences because they both have a 1 allele here and here, but this one has a 1 and a 2 and a 1 and a 2. So there are going to be 2 allele differences between A and B. And then if we look at the difference between A and C, let's see, we have a 1 and a 2, a 1 and a 3, a 1 and a 3, and a 1 and a 1. So this is actually the only allele it has in common. So the allele difference between A and C is going to be 3. Okay, I'm going to put up another little poll question. Well, I can't get the poll to work for some reason. So if you guys want to put your answers in the chat, that will work as well. No, 10 more seconds. Okay, it looks like everyone that put it in the chat got it correct, I think. So great job. Yeah, so this one's going to be 3 allele different because we have a difference here. That one's the same. We have a difference here and a difference here. So 3 differences. Again, all of this is usually done on, with a computer program. No one's sitting around counting these allele differences or anything, but just for understanding, it's kind of good to understand how we get to these results, I think. Okay, so we're going to go into a short little break. Okay, so we will start back up, picking back up with our core genome and whole genome MLST. We end up with allele codes through pulsenet as those set up. So it's going to be a hierarchical naming system. The benefit, and I think what's really cool about it, is that it shows relatedness between isolates based on the core genome MLST. It's going to offer a compact view of the entire population structure for an organism database. So they're set up for Salmonella, S-tech, Listeria, and Campylobacter jejuni. And then we must think about them in terms of population and not outbreaks, right? So they're distinct from outbreak codes. They can be used to identify clusters or help identify clusters, and as a form of nomenclature for referencing strains, which that's kind of one of the detriments of using the SNP-based analyses is we can say things are a certain amount of SNPs apart, but that gives us no way to name them or tell someone else about them, right? Or talk to another health department about them. But with core genome LST, we can do that. They can be complete or partial depending on how they relate on the tree from which it was built. And I'll give you an example of that. So here is an example code. So this is going to be the first 3 letters and 2 numbers are going to be the organism and version. So like Listeria monostogenes, version 1.2. The allele codes got updated recently. If you're looking at an allele code and it has 1.1 or something beside it, you'll know it's the old version and not the new version. And then this part here is the digits going to make up our allele code. And based on where the digits are, it's going to show how the allele difference range isolates that have matching digits in those spots. So this first digit is going to be 71 alleles. The second digit is 51, the third one 36, fourth one 19, fifth one 7, and sixth one 0. And they're going to be different for the different organisms. So don't memorize those for everything. So here is how they're set up for the different ones. The Campylobacter, see like the first digit, Campylobacter is going to mean 84 allele differences. S-Tech is going to mean 77 differences, Listeria 71, and Salmonella 80, right? And then they all end with 0. So if two organisms or two isolates have the same digits all the way down to the end, it's going to be that they are 0 alleles apart or approximately. So and then it's worth noting too that they all have 6 digits except for the S-Tech one, which only has 5. So in general, the more numbers in common two isolates have, the more closely related they are. They're approximate for all organisms, they're not exact, and we'll get into one of the reasons why later. Due to the clonal lineage of some serotypes or strains, the differences may be larger than you would expect based on the allele code, and you should always verify those with a dendrogram. Here's just some examples of allele codes, and I have like the example stereo one up here so that you can see the differences between the digits and refer back to that. So occasionally you'll see one without any digits, and it's going to be a singleton. It had no closed matches in the system, so it didn't have a name assigned. And then one day it may have one, but at this point in time, when you're looking at it, if it doesn't have one, that means there's nothing very closely related to it, or at least within 71 alleles. So in this example, this first one here, where it's 512, Listeria can have up to 6 digits, but this one only has 3. So it has a partial allele code. So it means they're a singleton in a cluster below their last digit. So for this one, it goes out to the 3rd digit. So it matches, it's closely related enough to stuff within 36 alleles, but it's not related to anything else between 36 and 0 alleles. So that's why it doesn't have any digits as of now. And if we want to compare these two to each other, we see that they share the first 3 digits. So if we look back up here, that means they're definitely within 36 alleles of each other, but they're not close enough to be 19 alleles. So it's somewhere between 36 and 19 alleles to each other. In this example, let's see, they share all 6 digits with each other all the way down. So they are indistinguishable or 0 alleles different based on their core genome. And then if we look at these top 2 ones here, we see that they share all 6 digits like the last one did. So they're indistinguishable or 0 alleles apart. But if we want to compare all 3 of these together, we see that they only share up to the 5th digit. And the bottom one's actually missing the 6th digit. So we know that it's going to be related to these first 2 somewhere within 0 and 7 alleles, or 1 and 7, I guess. So it's not indistinguishable like the other 2, but it's somewhere less than 7 alleles different than these 2. Okay, I have one more poll question. What is the allele difference range between isolate A and B in this example? Okay, 5 more seconds. Okay, so it seems there might be a little bit of confusion, so we'll go through this one real fast. So if we look at these two, they match up to the fourth digit, so they start differing from each other in the fifth digit. So if they match up into the fourth digit, that means they're 19 alleles, at least 19 alleles related, but less than 7. So it's going to be somewhere between 7 and 9 allele differences. And I have a question. Someone asked if there is a library of allele codes for various organisms that display the number of allele differences. So we can chat about this some more at the end. Jack or someone can speak up, but from what I know, and I have some screenshots later on, I think, that are from biogenumerics, but I assume there's something similar in PulseNet 2.0, where if you do want to look at two organisms or a dendrogram, it will show the allele differences in the tree. But yeah, if Jack or someone has a more specific answer about that, let us know. Recently, there's been an update with allele codes that was released with PulseNet 2.0. Again, same, they're for Salmonella S-tech, so not Shigella or non-S-tech E. coli, Listeria, and Campylobacter jejuni. They're now going to have unique digits at each threshold. So there will be longer numbers because they're using more numbers now. So like here's an example of the Salmonella one. So by the time we get out to fifth digit, it's 5 numbers long. And then clades for a specific threshold can be referenced by just looking at a single digit rather than the entire code. So you can, if you want to communicate with someone, you can say Salmonella allele code 11234 at the fourth digit, right? Instead of having to write out the whole thing now. So here's this example showing how the allele codes work with the previous version versus the new version. In this example, with isolate A, B, and C, they share the same fifth digit, but not the same fourth digit, right? So isolate A would not be part of the group because it has a different fifth digit, even though it has the same fourth digit. But with the new version, because there's unique digits, like any code with a 2 at the fifth digit is part of that group, right? So there would never be a one with a 1 there instead of a 2. So the group will share the same first, second, third, fourth digits along with the fifth. So that makes it a little clearer, and you can kind of look, you don't have to look backwards as much. So if they share the same fifth digit, you don't need to double check that the same fourth digit or third digit or first digit. And I just want to go through a few allele code limitations. Jack will probably kind of talk about this some more when we're talking about Cedric next week, but there's no allele codes for Shigella, so you can't use a cluster detection tool for this pathogen. Similarly, you can only use it for Campylobacter jejuni. You cannot use it for Campylobacter coli or Lauri or any of the other Campylobacter species. Certain search parameters you may use would result in a limited number of clusters available for viewing at once. So you have to narrow down the dates, the number of isolates, and allele thresholds. It'll be hard to see a complete list of these during your busy enteric season and may lead to missed clusters or outbreaks. So this is true probably in Cedric specifically. And you have to check each cluster each week to see if new isolates have been added versus looking at a dendrogram. You can just look at everything all at once. You may need to use NCBI, and again, we'll talk about NCBI next week, but especially when there's XPodes, because that may be a way you can kind of look at the distances to see what they actually are. Back to the question that was asked earlier, NCBI may be an alternate way to kind of find the actual differences. There's a few questions. Someone has noticed more X codes, yes, and we will talk about that in a minute. And is the allele code value just assigned sequentially when unique isolates are identified, or is it based on differences from a reference or something else? I assume it's sequentially, like as a new thing is pops up in that digit or that distance from each other, it would just be the next number up. So, okay. So an issue that came up a few years ago after they started using whole genome sequencing and they set up allele codes where allele codes and we're like merging and chaining. So that's going to lead us into the X codes. So here's an example. We have a red cluster here with some isolates in it. They are going to be up to 7 alleles different. And we know that because they have the same fourth digit. And then we also have this separate blue cluster here with isolates with up to 7 alleles different from each other, right? They have the same fourth digit. And if we were comparing between the two clusters, they only share up to the same third digit. So that means that they're up to 15 alleles different. The isolates in the red cluster are going to be up to 15 alleles different from the blue cluster, and isolates from the blue cluster are up to 15 from the red. But then maybe another isolate gets added that is actually going to make these two clusters merge. It's within 7 of the red ones, but it's also within 7 of the blue ones. So, like, what do we do, right? So that's when they end up having to set up the X codes. So when this chaining is what it's known as. So we end up with this merge cluster, and the isolates within this merge cluster are going to be 7 to 15 alleles different. But also, like the red ones, again, we're going to be less than 7, or the blue ones are going to be less than 7 as well. They're all going to share the same fourth digit, which would mean that up to seven allele differences, but they're actually going to be up to 15. So that's when they kind of had to institute the X codes. So X codes are going to be given when the difference is greater than about 4 times the expected threshold. So if it's less than that 4 times limit, it isn't or might not be given an X code. So some clonal lineages get merged together at a shared digit, or they're creating more differences than you would expect, just like that example I showed. So here's an example of an X code. You see it's just an X after the digits in the allele code. This is mostly in the Salmonella database. So Salmonellas is where you'll see a lot of X codes, and then sometimes with the STEC. So because of this happening, you cannot use the allele codes to determine the exact allele differences. So you would want to verify the allele differences with a dendrogram or a matrix, or you can use NCBI to look at if the isolates are closely related. So here is an example. And so all of the isolates share the first 3 digits. If we look over here on the right side, they're all 82.2.2. But then some don't have a digit or they have different digits after that. So based on that, we would expect allele differences up to 15. But the actual allele differences from this tree are 0 to 48. So they don't match, but they're not given an X code though, because it's not 4 times the threshold. Because if the threshold is 15, so 4 times that would be 60, but because the maximum difference is only 48, it's not given an X code. So that's why you still need to check them sometimes. And this is what I was talking about. Again, this is a screenshot from Bionumerics, so I'm not exactly sure what it looks like in PulseNet 2.0 right now. But in the dendrograms, you can see like it'll have this thing here, and this is the minimum allele difference and the maximum. So that's one place you could check it if that's still the same. Okay, someone asked me to repeat that. So if the expected allele difference based on the allele code is up to 15 alleles different, to be assigned a allele code, it has to be greater than about 4 times the expected threshold, right? So for the expected threshold for that one was 15. So for it to be more than 4 times that, it would have to be 15 times 4. So that would be 60. But because the actual difference is only 48, it's below 60, so it's not assigned an X code. And so someone also said that they notice enteritidis is a serotype that utilizes X codes more often than others. Yes, that is true. Enteritidis is a very clonal Salmonella serotype. So yeah, that's the place you'll definitely see them more. Another example, all isolates share all 6 digits, right? So without that X there, you might expect them to be 0 alleles different. But if you look over here in the dendrogram, it says the differences are 0 to 5. But it's going to be greater than the 4 times expected threshold, right? So if the expected difference is 0, 0 times 4 is 0. So since it's 5, it will be given a trailing XX code. Someone asked a question about the other one, so we'll go back. So in this example, they are being merged still. That's why the differences are greater than you would expect. So yeah, there are isolates within these different allele codes that have the difference that is greater. So they are still being merged, but how PulseNet has it set up with that 4 times the expected threshold allele difference, they are only given X codes if that's the case. So yeah, they're still merged and the distance doesn't match what it should, but it's not given an X code. And one more example. So all isolates in this example share the first 5 digits, 1, 2, 3, 4, 5. So they're just missing the 6th one. The expected allele difference would be somewhere between 1 and 4. And if you look over at the dendrogram, it says the differences are between 8 and 14. So they're not actually going to be a cluster if you're using a cluster threshold less than 5 or something. So that's why you always need to verify your allele differences, especially when there's X codes before you go on working on a cluster that might not actually be a cluster. Similar to the allele codes in general, X codes have been improved in PulseNet 2.0. They may continue past the X with plain numbers or additional numbers with an X. So before, when there was an X added, the allele code stopped at that point. So especially if it was added in an earlier number, that made the allele codes very useless. So yeah, so it's no longer truncated. And there can be multiple X's in the same code. So here's an example. Salmonella, we got 6778.3, 0.1, 0.1x, 0.16x, 0.16. So it has 2 different X's in it, and it continues after the X. Because of this change, we'll be able to detect smaller clusters of closely related sequences within the overall X code than you would have before. So showing you kind of what the differences are between the previous X codes and now. Previously, they were manually assigned monthly, and now they are automatically assigned, so they should be more up to date. And then before, if an X code was assigned at this 4th digit, there would be no more digits after that, right? So it's truncated. But with the new version, they're all assigned to any digit that's greater than 4 times the expected threshold. So again, if it's below 4 times the expected threshold, it will not be given an X. But it can continue past the X. So again, here in this first one, we see even though there's an X at this 4th digit, there's still a 5th and a 6th digit. And you can have more than one X in the same allele code if multiple digits are exceeding their expected threshold. So the expected threshold is going to be based on how the allele code format's set up. So with Salmonella, the one example we used before, it was 15, right? Because they matched up to the 3rd digit. So it's based on the thresholds that correspond to each of the digits. So that's where your expected threshold comes from. So if it is, say for Salmonella, if we're looking at the fourth digit to be greater than 4 times the expected threshold, it would have to be 7, 14, 21, 28, right? It would have to be at least 28. Or for this fifth one to be greater than 4 times the expected threshold, it would have to be 16. So that's where that 4 times calculation is coming from. Thanks for the question. Based on these updates to the Xcode, should be easier to find clusters of more closely related sequences with an overall Xcode, and it's more accurate than it was previously, especially since they're not truncated anymore. Anyone else have any more comments or questions to add about the allele codes right now? Someone said, should we interpret digits beyond an Xcode as having the expected threshold? As far as I know, yes, it's past that digit, it should be within the expected threshold. It's going to be at least less than 4 times, right? Because if it was more than 4 times at a digit past an X, it would also have an X in that digit. So it could be what it is expected, or it could be higher than expected, but still less than 4 times past there. The last content we're going to go into today is like thresholds. I just put these here. Y'all probably already have these. They're based on the case definitions, and they're different for local or national. So, 3 cases or 5 cases. And then they're based on allele differences. So 10 alleles for Campylobacter, for E. coli, and for Salmonella. And then for Listeria, it's either 10 or 25 alleles. And then all or most of them have a stipulation too, like more than 50% of the isolates must have to be within a certain number of days, or all of the isolates have to be within a certain number of days. And then for like the local Campylobacter, 2 isolates must differ within 5 alleles. So it kind of has 2 allele thresholds, one within the other one. If you notice, like I mentioned before, all of them are based on core genome LST, except for Listeria, which is based on whole genome LST. So allele differences within a cluster may be larger or smaller, depending on the organism and the epidemiological data you have. There can be similar strains or closely related strains that are determined by WGS that may not actually be epidemiologically linked. An example of that, like Relatedness, right? It's very clonal. You'll often find things that are very closely related, but they're actually not epi-linked. And then with zoonotic outbreaks, they may have larger allele differences than you're expecting. And that is because I like to give this example. These are based on actual numbers, and I've seen way more exaggerated differences, but I wanted to give some real numbers. If a flower outbreak, or max allele difference might be 4, and the isolation date range maybe from 5 months, so quite a while. And then this was from, I think, one of the live poultries from a previous year. And these are both the same serovar. These are both Infantis. But the maximum allele difference was 11, and the isolation date range was smaller, so 3 months. But if we want to think about like the biology of it, and that kind of explains at least a little bit why this is the case, is because if there is Salmonella living in flour or surviving flour, it's not going to be multiplying. It's kind of in a stress response. It's just kind of surviving in there. So it's not multiplying. So those dips and stuff are not happening because it's not replicating. Whereas in chicken, the Salmonella is, kind of happy. That's where it wants to be. It's a warmer temperature. It's a more moist environment. It's going to be replicating a lot more rapidly than in the flour, right? So you would see more SNP or allele differences in a shorter amount of time compared to the flour. So that's one reason you need to take into account like anything about the epi data or the sources that you know of to maybe adjust your thresholds to include more cases or to exclude some cases to actually get them to what they should be. for this outbreak or this cluster that you're looking at. So yeah, you have to think about those things. And then I know often people talk about Relatedness and stuff. They use a smaller threshold, and for other serivars, they might use a larger threshold. We don't have a perfect system figured out exactly for what it should be, and we have guidelines for salmonella just in general, but ideally in the future we would have different thresholds for different serivars or different circumstances. And I think that would be something interesting to discuss now if anyone wants to talk about what different thresholds you use. So before we do that, I was going to tell y'all about the next two sessions. Next one's going to be next Tuesday, and we will cover rep codes and documenting and investigating local rep strains, NCBI pathogen detection and how to use that for clustered investigation and new cases. And then for Cedric, using it for cluster detection and investigation to find other cases, to look up historical information about rare serotypes. And then session 3 would be in 2 weeks on July 1st, and it's only going to be 1 hour. One of my colleagues at TDH, Mukta, will be showing some Tableau dashboards and may talk a little bit about StatScan. And we'll talk about communicating WGS results to stakeholders and kind of summarizing that so others can understand, and standard phrasing that CDC uses. And then she'll talk a little bit about PulseNet 2.0 and how to get access to that. So I will open up the floor now. You can ask questions in chat, you can unmute yourself, or if anyone wants to discuss something, I will answer a couple questions that are in the chat. So a person can interpret there is at least as many SNP differences as allele differences, although it could be many more SNPs apart than alleles apart. Isolates that are 3 alleles apart or at least 3 SNPs apart, but it could be greater than 3 SNPs apart. So that is something that you cannot know for sure, I think. And it's because, like I mentioned earlier, like you may filter out lower quality SNPs or things. So the SNP differences may not translate the same. Some programs, when they're doing SNP differences, they will mask out parts of the genome, like phage genes or accessory genes or things like that. So they may not be included. So it's hard to say whether the SNP or allele differences will match up or how much more or less they might be. I would say that yes, they would be roughly, and or that they would be comparable. So if you had like 3 isolates and 2 or 0 SNPs apart and the third one's 20, right? And then you look at alleles and it's 5 alleles apart and the third one's 30, right? Like they're gonna usually be in the same, everything will be relatively the same, at least, if that makes sense. Okay, someone wants to go to slide 56 and walk through some of the interpretation of the X codes in the tree shown. Okay, so this tree actually does not have any distances on it, so I can't say anything about what the actual distances are, though. So, yeah, I can't really say much other than, like, if you look at these, then we know that the distances at, let's see, 1, 2, 3, 4, 5. So the 4th and the 5th digit are going to be at least 4 times greater than they're expected to be. So I think the 4th digit was 15, right? If I remember correctly. So that would be they're up to at least up to 60 differences. And then this one, I think it was 4, so they're up to 16 differences. In those digits. That's all I can really say from looking at this, because it doesn't say what the actual distances are. I'll say, Jack or anyone, I think you answered the question. Is there not a way to see them in PulseNet, the actual distances?
You should be able to find actual allelic differences in PulseNet. I know with PulseNet 2.0, depending on what isolates you're looking at, you might have to get a little creative with some of the filters, or you can just kind of add things to comparisons if you need to use different filters to find different isolates. But Yes, PulseNet, you should be able to, if you can get the right isolates, you should be able to compare them and get allelic difference.
Okay, because I assumed there was some way, like with these screenshots from bionumerics, where you could see like the minimum and maximum allele differences. There should be some way to do it, or maybe there's a scale that's cut off in a screenshot somewhere,
If the bars are, I mean, if the bars are vertical like that,
like
but yeah,
here.
it's a little hard to tell if they're what is fully vertical and what is slightly not fully vertical.
And that's the whole
But I
scale of it, right? It could be like if if this bar is 500 SNPs, like even if they look vertical here, they might not be. But if this is 5 SNPs, then these are probably zero, right? Or not SNPs, sorry, alleles. But yeah, I think it depends on the scale. Okay, is the unit for the allele a full gene? So they call them loci usually, which is it includes genes, but I think it can be other things as well. I'm not 100% sure on that. I don't remember, but in practical terms, it's usually like a gene, though. Okay, someone says you can display allele differences in PulseNet 2.0, but when isolates are super close together in the tree, it can be glitchy and not show up. But there's also an allele matrix option. Okay, perfect. So you should be able to, if there's some Xcode ones you need to look into more or something, you should be able to pull up the actual distances so you can actually compare them. Does anyone want to chat about how they've what kind of adjustments they have made to their thresholds for certain salmonella serovars or and questions about that. So again, I think that's something people often are asking about or don't know or have good information to share. I think it's a good idea to start small and then broaden your window, yeah, if you need to. And I know like in Tennessee that especially with enteritis, like in a slow time of year, they might look into 2Z clusters, like, but like during the busy times, they may or may not even deal with the 2Z enteritises because there would just be a lot, probably. Definitely agree. Important to look at both the local and the national level. But yeah, you got to like pay attention to what organism or serotype it is as well, because you may need to adjust your thresholds or, you know, the timing of things as well, I think, too, because like, some of those frozen food listeria outbreaks, if you were only looking at 30 or 60 days, you would never have caught them because the windows for some of those food items are very long. And same with maybe like the salmonella in flour, right? If it's can survive and salmonella can survive in low water activity foods for a very long time. So in theory, they could be kind of spread out. We have 3 more minutes, and you can have one or two more questions if anyone has anything. Otherwise, we will see all or most of you next week, and then the week after for the next two sessions. And always, as always, feel free to reach out to us at the Tennessee COE if you ever have questions or need anyone to, double check something or, I think we could like we're available to do like consultations and stuff as well. So, Emily, if you can you put the TNCOE email in the chat for us? Say like, it depends on I think a lot of people I've talked to kind of up or down their thresholds or their number of case thresholds based on their manpower, right? Like if you have your enteritis threshold set at 10 and you're getting hundreds a week, like you can't do that probably. So you may lower it to 5. But if it's, if you got time and people, then you may up your threshold to 15. Well, I'm gonna look. I think someone might have done some studies on this. I will get back to you if I find it, but I feel like someone did something where they kind of looked at actual outbreaks and compared the thresholds back to it. But I will get back to you if I can find that. And then Kelly, do you have an opinion? Because you probably know more than I do about this.
Hey, yeah, I actually have been answering very similar questions just about different pathogens and serotypes. And pretty much, I would say prioritize anything between 0 and 5 SNPs. That's usually what I think. I know the study that you're talking about, and that's what they reference in terms of like
Mm,
finding outbreaks um, and epi data, like linking the isolates that were between 0 and 5 SNPs. Anything more than 5 SNPs is like a little bit like sketchy, I would say. Important, but not like super urgent. And then anything greater than 10 SNPs, it's more like curiosity. So that's kind of what we've been using for Salmonella enteritidis. We definitely want to prioritize anything between 0 and 3 SNPs. That definitely is like a red flag for us, but we would consider adding things up to 5 SNPs if it's like an outbreak or something. And the epi data like makes sense.
thanks, Kelly. That was more helpful. Well, again, if anyone had any more questions, feel free to email me or the Tennessee COE, and we will see y'all next week. Bye, everyone.
Bye.